All right, I'm going to kick things off. Welcome to Unit Testing. This is a live conversation series we host at the intersection of deep tech and frontier science. This is our third installment, episode number 3. My name is Kyle O'Brien. I'm a founding partner at Unit Ventures. We are an early stage deep tech fund based here in Paris, France. And I'm really thrilled to be discussing a topic that is near and dear to our hearts at Unit, but I would imagine all of yours as well in the audience, which is that of aging and longevity research. And we've got a few amazing guests today, and I'm going to turn it over to them to do some quick introductions. So to start, I will allow my colleague and partner, Rand Hindi, to give a quick background on himself.
Hey guys, my name is Rand. My background is actually AI applied to biology. And after that, I was running an AI company that got acquired, and now I'm running a cybersecurity cryptography company. I also do quite a lot of investing with Kyle. So my kind of sweet spot, what I get excited about are usually very early stage companies building things that most investors don't understand, right? And so, you know, having been at this intersection of like bio and AI for 20 years now, I've always been a biohacker in my kind of spare time. So, you know, I like to joke that what I do on weekends and evenings for fun is reading medical and bio papers to try and see what new stuff I could try on myself. And so, yeah, so super happy about having this conversation, super excited about it. And as Cal said, you know, this is an open conversation. We're keeping those events small on purpose so that we can have like this kind of feeling of being like in the same living rooms chatting about this topic as opposed to like a conference. So anything you want to talk about, anything you want to contribute, please feel free to jump in and do that.
Great. I will hand it off to Nina, the CEO and founder of GlycanAge. Nina, take it away.
Hey, so my background is actually not in science. I was in tech for 12 years, but I come from this very interesting family of scientists. So my father's a glycobiologist, my mother's a neuroscientist, and he kind of, mainly my father, he dedicated his life to the space of glycobiology and then stumbled onto an aging clock about 10 years ago. And this was one of those first, actually Horvath's and our glycan clock was published at the same time. So he thought that this could be something that people can use, but there wasn't real utility for it about 10 years ago. So the first idea was forensics. You know, can we tell the age of a sample? But then we realized there's more utility and the longevity industry came up. So I crossed over to bio about 4 years ago.
Very cool. As hard as you tried, you couldn't escape the family business, it seems. I think they tend to pull you back. All right, Max, I'll let you take the mic.
So hi guys, I'm Max. I'm a scientist by training. Currently finishing up my PhD in Singapore on the combination of like AI, complexity science, longevity, on the intersection. And my career part was a little bit, I first went into biophysics, then got bored of biology, went to complex systems, which led me to AI. And now I'm doing like the first full circle back more like towards AI and physics to longevity. But apart from being a scientist, I also like wander a little bit in the business world, being mainly involved with VitaDAO, where I run events across the globe to promote longevity, increase the audience, especially talking to people that don't know about longevity yet or heard about it, are curious, because we just need more people in that space from all branches of life. Health is important. A non-healthy population costs money for the government and for everybody else. So it's almost like this, we need to organize in new structures and educate each other and help each other to get to better life extension, health extension technologies.
Fantastic. Well, we flipped a coin backstage before this, and it looks like it landed on Max for giving an overview of the health and longevity space. I would imagine this is going to be difficult to sum up in a short 3 to 5 minute monologue, but evidently you are the right man for the job. So maybe just as a primer for the conversation, you can give us a potted history on what exactly is longevity and aging research, and why is it important today?
Sure. So it was a biased coin that was flipped. So I think depends how long you want to go back in history, right? I mean, some of the first texts in the world that were written already dealt with like histories of like the fountain of youth and immortality, right? Back then, maybe less science-based, but like more hopeful. But I think the spirit probably lives now along, like lives within scientists and a bunch of other people. But I think the first real experiments that concluded that lifespan can be extended was like roughly 9 years ago in 1935. It was fairly simple. They just starved rats or like caloric restricted those rats, and they just lived like 33% longer. And that in a way kicked off like a lot of those caloric restrictions theories or interventions that we like, funny enough, like 90 years later, we still don't have like the clear data that says caloric restriction is always good for you. It's not always good for you, right? So 9 years later, it's still like ongoing research. And I think then fast forward like to the mid 90s, a bunch of smart scientists found that you can use like genetics and modulate lifespan of worms and yeast and other organisms, so that there was like something in a way within our genetic code that we can use to modulate the biology of aging. And then further down in the 2000s, there were like a few molecules that have been found that were like very significant. Rapamycin, the most promising on that one, which like comes from a remote island, the Easter Islands, and was like dug out of the soil from a bacterium, and somehow it's a long story. It even got frozen. It was almost thrown away, and then they somehow found out that it does stuff, and that's extends lifespan in mice by up to 30% in every other organism tested. And that was like really the sign, okay, we can use small molecules to modulate aging biology, and get like maybe a hold of longevity. That was back then like still mostly like academic, but I think then like from the 2010s 2015 on, you slowly had like companies being like shaped and formed, trying different approaches to get a hang on all those things. And like now in 2020, we have like this entire burgeoning industry where we have obviously like aging clocks, like statistical models applied to biological data to predict what is actually your biological age, what is your aging rate. And then on the other side of that, obviously biotechs and supplements that seem to have an impact on how you're aging, on how you age, and potentially even rejuvenation in the future.
Great. Well, so I would consider myself
before getting to that, I have a question, Max. Sorry, just a question. So, you know, we often kind of like see studies where they can extend the lifespan of mice by like 10, 20, 30%. So I just have two questions. First, why is it always like kind of capped at like 30%? Like, why is it never like we double the lifespan of a mouse? And secondly, what happens if you combine all of those things that are supposedly adding 30%, right?
So that like multiple ways on that. I think in somewhere in the mid 2000s something, there was actually a prize called the Methuselah Prize, who was like said if somebody doubles the mouse lifespan, they will get like $1 million or so. I think this prize still is open, and nobody has gotten it so far. And I think currently why most of those interventions plateau is many of them, until recently, I must say, were like small molecule interventions. They probably all, in one way or another, targeted the nutrient sensing network. So they were like all, in a way, calorie restriction mimetics or something along those lines. So they were like all targeting the same things to a large extent, maybe a little bit senescence as well. And now, yeah, people and the thing is, those studies are expensive, right? So usually people only like test like one compound, and then maybe like different dosages of that. But they wouldn't do like 100 different compounds. Even people probably should do that. And you have like the intervention testing program, which looks a little bit more into that they have like a large set of mice, and they every year test like different molecules to see if it's at least reproducible, right? So at least like we know that some of those interventions can be reproduced, some are not. But what you're talking about is like combining therapies. People started looking into it more and more, and the idea is obviously, like I said, if one is adding 20%, the other one should be at 20% as well, and maybe there's like a super synergistic effect. And that seems probably true for some of those combinations, but not all of them, right? So some of them, they don't add up because they target almost the same thing. Whereas like the ones that might, ideally they would be orthogonal, right? They would be like hitting entirely different pathways, and they should be adding up. What is interesting, I've recently saw some promising data of like a combination of like not only small molecules, but they really went into small molecules, gene therapy, stem cells, and combined it all in mice. And the data there looks very promising. It's like from the Longevity Escape Velocity Foundation. That's like the new foundation that Aubrey de Grey spun up. And he's posting about those results on Twitter and Facebook. And I think that looks really good because you see like small molecules alone do what they're supposed to do compared like to controls. But if you go further along with the combination, it even works better, right? And that's very promising results in mice, I would claim. And yeah, more people will try that, should try that. So there's like a lot of room for improvement and tinkering with combination therapies.
Okay, super interesting. Okay. I guess, I mean, that could be a good segue into Nina and what you guys are doing, right? Because I think we talk a lot about extending lifespan as in when you die, right? But it's kind of useful to know when you'll die as well, right? So like having some kind of like measure of where you are in your own sort of like biology is something that would be super useful. Like if I'm 70, if I'm biologically 70 today, I better do something about it now, right? And so
Yeah.
can you tell us a little bit more about those clocks and the stuff you're doing at GlycanAge and, you know, your vision?
I think you're talking more about the personal application of them, but the first utility was actually in research, because these longevity studies, they take a very long time. Like if you wanted to do a longevity study in humans, you know, it would be half of your lifespan at least. So we need these surrogate biomarkers to tell us if we're heading in the right direction or not. So the first kind of utility of the clocks. But then also there's a personal utility, because we all, like, we're individuals and we're unique by genetics, epigenetics, environmental influences, and the same thing will never work for everyone. So even when you have a successful trial of a small molecule, maybe you see, if it's a good drug, maybe you see above 50% benefit, but then a large chunk of people won't benefit. So I think here you have a lot of good candidates. And also there are going to be eventually very different targets for longevity. Some could be maximum lifespan. The other might be time to disease, and some might be more, how would you say, skin aging or so. Something that's less connected to health, but is impacting an aging process. So our one is looking at glycans, and there's maybe two candidates which are now used more and more in trials, which is epigenetics and glycomics. And very rarely do you see them respond to the same thing, but there is some commonality. Like recently we had a rat study where it's kind of like a young, taking plasma. Actually, in this case, it was from, what is a nice name for this? Young piglets going into rats. And they looked at both. This is kind of like taking young blood and putting it into an old animal. And in both cases, our glycomic clock and the epigenetic clock reduced by 50%. Now, this wasn't a control study, needs to be replicated. There's a lot of good studies already on this young plasma. The problem as a therapy, it's not very, until we figure out what are these small molecules in young plasma that influence longevity, we don't really have a therapy that you can apply broadly.
Can't you amplify plasma to like build more of it from a sample?
Can you repeat it? Can you do
if you have a sample, like, you know, the same way that you can take some stem cells and replicate them to have a bunch more, could you do the same with plasma and young blood? Or do you actually need to draw the blood that you're, you know, redoing? I
think at this point you need to draw the blood. I don't know if Max has any input on.
Yeah, I think I think artificial blood is like not solved yet either, right? So it's always the question: could you like just like, I mean, otherwise you wouldn't have even like blood donations for humans, right? If you could like just like create infinite amounts of blood, that it's actually a big problem.
Interesting. Okay. Yeah. Who's working on that?
I think some people are. I don't know anybody though.
Like an important problem to solve, no?
Absolutely, yeah. That's
Okay.
I agree.
Yeah, I can add something interesting from, so we're at this longevity week around the Bay Area, and Steve Horvath had a talk yesterday, and he was looking at, he has this clock that's very good at predicting mortality. He has this clock that's very good at predicting somebody's chronological age. And then he developed a clock that predicts maximum lifespan in all kinds of mammalian species. And the clock that predicts maximum lifespan did not correlate with the predictor of time to disease or all-cause mortality. So there will be, aging is such a complex challenge. It's never going to be one thing. And we die far sooner than what our maximum potential is. So there's going to be these different mechanisms we need to tackle to get to different milestones of longevity.
Interesting.
So I thought that was super interesting.
Okay, so the idea is that you have a clock that tells you how long you could live if you did everything right, right? And then you have a clock that tells you if you're doing everything right. And basically that's like, okay, interesting. And how genetics, how much genetics is into your maximum lifespan versus epigenetics, for example? Like how much is environmental? How much is like you're born to live to 100 or live to 120 or to live to 80?
So I don't think he showed that data, so we don't yet know, but probably he can do this experiment. We did it on our clock. So with glycans, you're mainly, or the ones that we're looking at the moment, you're mainly looking at immune aging or a process called inflammaging. So this is the theory of aging of damage throughout life. You want to minimize this damage. And there we looked at heritability in 3 twin cohorts. It was about 40% genetic. So a large part is genetic, and then the other remaining part is epigenetics, environment, and that's something you can modify. Why one is a lottery.
Do you think you could create like some kind of like CRISPR gene therapy to literally change your genetic lifespan in that case?
At one point, yes, there is a recent paper in mice when they identified these longevity genes and then developed a model for that. And I think they all died prematurely. So it's such a complex system. Like it's not going to be one thing. It's not just going to be genetics. You're going to have to kind of unlock all these different layers where, glycans play a big role, lipids play a big role, and so forth.
Interesting. I mean, it's quite dystopian to imagine that like maybe people are gonna like start having babies together, by looking at how long each of them would genetically live, right? Like you could imagine like, same way that people are like, oh, we've got blue eyes, for a baby will have blue eyes. We're both gonna live to 100, so a baby should be living to 100 kind of thing, right? I mean,
I think here you have these two, not conflicting theories, but I guess they can go together. One is this aging is a program. So you have like a maximum program, and we can reprogram that system and get life extension. But the other one is law of damage, something that you have to look after, control throughout your life. So in a way, your genetics don't matter if you're going to destroy your health through. Yes. So
can you tell us a little bit more about inflammaging and glycans and everything? Because I think for most people, this should be fairly new as a concept.
Well, glycans are, as a concept, even in the scientific community, there's still a lot of education remaining. So maybe we start from glycans. So if we look at the cell biology, we have 4 components, which is DNA, protein, lipid, and carb. And carbs are also glycans. Now, the problem is when you think of carbs, as individuals, you think of the carbs you're eating and maybe how your biology is responding to that. This is also how physicians think about it. What we forget is that actually these carbs evolved as a language for our biology to communicate. So they're this kind of like last step in evolution, which enabled us to be multicellular. So that's the type of carbs we look at. So you have all of these key processes where carbs are involved, glycans are involved, you just don't know them by name. One is blood groups. So glycans in your blood cells would define your blood group. Your immune system would largely communicate through them. For example, cancer would disguise itself in these healthy, good-looking glycans to evade immune surveillance. And even conception happens because of glycans. because it's a foreign entity that covers itself in good glycans and evades the woman's immune system. So they kind of play a key role in all these processes since we were multicellular, but there's very little understanding of them. Where we understand them is the immune system. So this is largely all of our research focus for the last 30 years. But you also have them on lipids, you have them in so many other places that we just haven't yet explored. But when we look at them on the immune system and we look at aging, we mainly see that on the adaptive immune system. And there it kind of changes the composition of glycans on your antibodies. You would have more of these anti-inflammatory ones when you're young and less of these pro-inflammatory structures. And then this composition would change where that shifts. So you have more of these pro-inflammatory ones as you age and less of these anti-inflammatory ones. So this is the inflammation theory. But then this is a core mechanism of most of our diseases. So an overactive immune system is autoimmunity, but a lot of cardiometabolic diseases have this like sterile, slowly accumulating inflammation as a key mechanism. And cancer is your immune system not killing something that it was very good at when it was much younger. So it's a core mechanism, majority of our disease. And what's unique about glycans, because there's something in between your genes and what's happening to your biology throughout life. So they incorporate your genetics, epigenetics, and your environment. They can really nicely tell us early on this time to disease. So if we look at particular glycan signatures, we can see up to 10 years in advance that you are heading towards an autoimmune condition or cardiovascular disease or diabetes. And they're not genetically fixed. So they would, yeah, sorry, I can hear you.
You're saying you can predict 10 years in advance
what
will happen. Wow, okay.
Yeah, so this is all research cohorts. You have people when they're healthy, you follow them for 10, 20 years, and then you see how their glycan patterns would change. And we would see these changes up to 10 years in advance. But even more interestingly, they respond to intervention. They're not fixed. So what we see would predict hypertension responds to physical activity. And what we see would predict insulin resistance or diabetes responds to caloric restriction or weight loss. So the difference here, of course, these are all things we know we should be doing. You know, don't eat too much and exercise. But it's a little bit different when you have this feedback 10 years in advance. So you can tell you, hey, you will develop that, you know, you're heading towards diabetes. It's time to lose weight now. It's time to do this hard lifestyle change now before you have a problem. Because what happens today, you get a diagnosis. You go in for a blood test, and the only point that you'll identify the issue is when you already have the hypertension or you have diabetes. Before then, nobody's going to give you a warning for it.
It seems to me, and Max, I don't know what you think about that, but it seems to me that like a lot of like the very promising longevity stuff happening is around the immune system. Like almost as if, so I guess forget if you're like 90 years old and trying to rejuvenate, right? Just assuming you're like in your 20s or 30s or 40s and you just want to stay there. Isn't it just about the immune system? If your immune system works, it kind of like takes care of like everything. It clears out your senescent cells. It, you know, repairs everything that's broken. It kills cancer. Like, do you need anything else than a healthy immune system when you're like young?
So I would say in biology, like everything is connected, right? And only like trying to fix one thing will probably still not fix all of the problems, right? But I agree, like the immune system, it's like systematic, right? It's like everywhere in your body, and having a healthy immune system certainly is important for like all the reasons that you just mentioned. And I think when you look at it, like just last week, they announced the X Prize, right? This like $100 million prize. And how they separated is like you need to develop a therapy that works in the immune system, on muscle, and on the brain, right? And maybe that's like a different way of looking about it, right? You can still probably have a good immune system, but you could be frail, right? So you could so that's one thing, right? That's might not get any diseases, but you just don't have any power, right, to do anything, which is probably not so good. And then with regards to the brain, given the and I must say I'm not an expert on that, right? Given there's like the blood-brain barrier, there might be some part of the immune system that are different in the brain than it is in the body, right? And we know that obviously, well, not obviously, but a lot of people think that a lot of mental health issues are actually kind of inflammation in the brain as well, right? So there's like there might be like some other immune response going on, right? So maybe separating in those three things. But then you could also make the argument, well, the immune system is like obviously like all like your adaptive and innate immune system, right? And it all comes down to the cellular level, right? Where you say, well, if you just could keep your immune cells healthy, that would be good enough. But then you directly again at, okay, let's keep the mitochondria healthy, let's keep the DNA stable, and keep the nutrient sensing within the cell, okay, and then it all connects again to each other. But when you say, well, okay, let's keep the mitochondria in the immune cells healthy, why not directly keep them healthy in all the other cells as well? Which like just shows like
yes,
I think
I suppose
really
I mean, if the immune system has the innate ability to fix everything, fixing the thing that fixes everything supposedly should fix, mitochondria and muscle. It should fix, brain inflammation. Wouldn't all of that like kind of just waterfall from, a good immune system?
maybe I'm not fully sure. I'm not sure how a good immune system would, for example, fix DNA damage that would create cancer, right? It will eventually take care of the cancerous cell, but it wouldn't like modulate it like on a molecular level within the cell.
Can you share with everyone here, what are like the main ideas to achieve, like longevity, right? So like what are like the big, sort of like verticals that people are currently working on?
So there have been like a few, the theory of why we age has changed over the last decades, right? Currently the I would say somewhat a consensus is that like 8 or 13 or 9 hallmarks of aging, and that they're evolving as well, right? And those are like all and that mainly relates to mammalian aging, and it's like your mitochondria, your DNA instability, telomeres going dead, nutrient sensing, stem cell depletion, extracellular matrix, your changes in your microbiome, the other 4 I currently forget. So it's like mainly like specific processes that if they like go wrong, or like something in them changes, will produce the aging process. And then the idea is that, well, if we could just stop them, or stop those processes, or find therapies that directly target those processes instead of like disease, then we probably would prevent disease, right? So the big assumption is that like aging is the root cause of age-related diseases, and without aging, most of those diseases wouldn't occur, right? Most of the time, like you get all those diseases from 60, 65 onwards, right? They're usually like not 15-year or 20-year-old people that have Alzheimer's disease.
Right. So you don't die of like a disease, you die of aging, which basically made the disease exist in the first place.
Correct. That's in most cases, right? Like in biology, a few abnormalities where young people get certain diseases, but that's usually what happens. You get old and then you get all those diseases. And the idea is if we could just like delay or reverse or stall the aging process, or even slow it down, all those diseases would occur later or never. And then the good thing is if you would like in a way compress morbidity, like that you wouldn't have any of those diseases, life would be much better. And ideally you live longer.
Okay, so that works for us, right? Because we're young. But what do I do if I'm already in my 70s, 80s, and 90s, and I just don't want to die?
Well, I don't know. It seems like a lot of people become more God-fearing when they get older, right? Maybe that's a coping mechanism. But I mean, as bad as it's other thing, when you're 70 and 80, there are like not that many things you currently can do, right? I mean, there's like all the things that your doctor tells you, well, don't eat healthy, have good sleep, have good rest.
But how about like all of those rejuvenation stuff, right? So like, organ replacement, reprogramming, like they're supposed to like reverse aging. So that should work for someone older, no?
Yeah, sure, but you're currently 80. I think until those things work, you might not be around anymore, as bad as it sounds.
Oh, you
And the
think?
problem is
That long,
with,
huh?
yeah, I mean, if you look how long it takes to develop medicines, right? And that's probably one big problem of the FDA. Like it takes 10, 15, 20 years to get some of those things to market, right? And I mean, reprogramming currently has like a lot of support, a lot of momentum, a lot of hype. But that is like still scientific experiments, right? A lot of that stuff. I mean, most of the mice, they get some kind of cancer. They don't live long enough. Having that in vivo as a therapy is probably still further out. I think what is probably like the most promising approach right now for reprogramming is like, for example, taking your immune cells out, rejuvenating them, and then implanting your immune cells again, right? So it's like that, and I think that's like something valuable in that, but even that is like not fully solved, because the problem is if you have like 10,000 cells and 9,999 of them are fine and reprogramming work, and this one other one failed and now has like some cancerous properties, you probably don't want to have the cell in your body. So there's like still like a lot of problems to be solved. I think it's super exciting technology. It's super cool to study. But it's like not prime time for that yet to be rolled out into humans. And I would assume this will take like another 15 to 20 years until that goes to market.
Okay, that's not so bad, right? I'll still be in my 50s by then.
But okay, there might be like a large error bar. I'm attaching a large error bar to it.
Okay.
So I want to interject with a quick question from the audience, and I think maybe to preface, Max, you can tell us a little bit about some of the lipid research we were discussing prior to the call. But a guy in the audience named Tom asked, what is the most impactful thing that an average person can do to help accelerate progress on age reversal research, longevity? I would imagine in your position at VitaDAO, Max, you do a lot of evangelism here. You've probably seen all the various categories of research. What would you suggest to someone who's not a PhD but is interested in the subject and wants to get involved, both for their personal benefit and I think for the bigger picture for the world?
So, I mean, I always guess it depends on what your skill set is. And I think as in any industry, every kind of skill set is needed. Like, I mean, sure, you have PhDs and they do their science, but a lot of them have shortcomings in communicating their science. And so it's important that we get better communicators out there that come from a non-science background, but are interested in it, just curious, and start talking about it. But then, even then, I mean, you need business people, business-minded people who help a lot of academics that have good ideas spin out those companies and say, hey, well, you do your science. I think it's promising. Let me help you with all the business side of those things, of like developing the therapies or biomarkers or whatever is cooking in the lab. And then I think probably one of the most impactful things is like just talk to your friends and your family about it and say, hey, how do you think about it? Ask them questions like, hey, how long would you like to live? And what does it mean for you to live healthy in old age? And maybe in a way put like some thoughts into their mind of how healthy aging could be, and that this currently thinking of like you grow old and you grow sick might not have to be the future anymore.
About the question for the audience, guys, I mean, maybe to make it a little bit more open floor, if you have a question, just unmute yourself and maybe ask it. Should we try that?
Yeah, go for it. There's a couple points from Harold and Francois about AI and quantum accelerating speed of innovation, and some discussion around other species, plant species, for instance, living thousands of years. Why can't we? Either of you want to jump in?
I can take the AI one. I can't speak to quantum. That's not much in my world. But I mean, if you assume that AI is currently almost changing every industry, you should probably bet that AI is going to change biomedicine and biotechnology, and thereby will absolutely impact longevity. And then and the thing is, a lot of AI people, software engineers, trying to get into longevity and want to do that. And there's like currently like no clear path, because it's like this vast venue of like different approaches. And then I always think in a way that like the last 10 years, AI was like mainly in this like coming from like this digital space where a lot of data was available through social media, through imagery, to like the written word and language. And obviously the promise is that like AI will like bring like similar acceleration or breakthroughs to biology. And I think it's the right idea, but I think there are like certain problems that we have in the biomedical space. A is like the availability of data, and that goes in a way that how data is generated in a laboratory is, in most cases, it's like not those large quantities of data. It's like maybe like 100 mice, and they run an experiment on that, and then you can analyze it. But for that, you don't need your fancy deep learning algorithms, right? This is like old school statistics in a way, and that's powerful and good, but not what people currently would think of if they say, "Hey, I want to do AI for longevity." At the same time, there are like more and more studies coming out that have like larger amount of mice, and maybe that's being useful. And then there's obviously more and more human data available, right? That people like over the last 40 years, there have been like cohorts being assembled for tracking populations with 30,000-40,000 people, and now they all have the blood stored, and now all this blood is reanalyzed with like the latest technologies. Be it like glycans from the lab that Nicolina works with, or like lipids in the lab that I'm involved with, or methylation, or other things. So there's like more data available that can then be really harnessed for like those AI things, or even like medical records, right? Which is like probably the most precise collection of medical knowledge that we have.
Can we talk a little bit about actually about like data modeling, because and maybe this is more for Nicolina, I don't know, but I read a paper recently, and I'm surprised that paper didn't get shared a little bit more, but I read a paper recently that was showing that with the little amount of actual individuals in those epigenetic studies, it was very likely that the clocks that we were using were just, basically just chance correlation, right? And they were showing that there's like some ridiculous experiment, but they could show that like they could predict from epigenetics with like a 95% correctness, like your zip code in the US, like something stupid like that. It makes no sense, right? I mean, I guess you could say it does make sense with pollution or something, but it should not make sense, right? And so the logic here was that we might actually be deriving clocks by just blindly throwing a bunch of data into like some kind of machine learning system, and it produces a model, but really that model doesn't mean anything biologically. Like, what do you think about that?
So I can add to it, and I also want to add some things on the different species and plant, but I think the problem with epigenetics, you can model a lot of things on that. It's a huge data set. You're looking at this huge information set that we only understand a little bit of. So, for example, the mortality clocks are heavily weighted on smoking, cardiovascular risk, things like that. And the original epigenetic clocks are heavily weighted on age. So they're very good to predict chronological age. They're not as good at predicting time to different health events. So that's why you have many different epigenetic clocks. If anything, there's probably about 20 different ones at the moment. And you need to really, if you're doing an experiment, we need to really look at closely what one might fit what you're doing. But then there's very little data showing responsiveness to intervention. And one reason for that is the noise, because they're looking at many things. This reproducibility of a result is not so straightforward. The first versions, they had a 10-year error margin. So if you test the same sample twice, you can get 10-year biological age up and down. Now, the better models are about 3.6 years error margin up to 10, depending on which clock. So the stability issue is a big issue in the epigenetic space. We don't have the same problem because we're literally looking at this one key, this very antibodies are some of the most abundant proteins in blood. And we're looking at just the antibody glycome. And the method we developed is very precise. So if you test the same sample twice, the variability is less than 1% or a year in biological age. So if you see a shift, it's an actual biological shift, even if it's a couple of years.
You can also explain why.
And you can explain why.
So the epigenetic clocks, if you look at the CPG sites that are used in the models, like you can't really tell why those specific, like sites are actually meaningful, right? So that's why like it's difficult, it feels a lot like the early days of machine learning where people would just build models of everything and they would have no clue why they work. It was kind of like a black box kind of thing. And, in some cases it did work for a long time, but in many cases it just didn't work in practice because it turns out that it was just spurious correlation, right? So can you actually meaningfully use a biological clock if you don't understand why it predicts the way it predicts?
So if you ask Steve Horvath now on the scientific utility is clear. I think it's a very exciting space. There's a lot of great discoveries. It should be applied more in many ways in this Asian research space. But consumer-wise, if you ask Steve Horvath, he'll tell you not to waste your money on the epigenetic clocks. First, there's many different kinds. A lot of them are just derived from his first version, the Horvath clock. And the reason a lot of new clocks came up because there was a lot of, it was very good at chronological age. It didn't have as much utility for other things. So I think consumer-wise, we're quite early. And I think that each of these clocks will have different, just different epigenetic clocks are going to have different mechanisms and then maybe even different interventions that they'll respond to and so forth. And just a little bit more work needs to be done in this space.
It's really tough, to, as a consumer, at least, like as a person, to find meaningful, stuff because with that, like, I think I could consider myself to be like a biological clock nerd in the sense that I love measuring, all kinds of clocks. And, every 6 months or so, I'll go through like a full checkup and I'll compute every single clock I can. Like I'm literally the guy that will download the raw epigenetic data from one of the tests and put that into like some, statistical package to compute the other clocks. The range, like the spread of age between those clocks is insane. I think my youngest age is actually with you guys with the glycan age. I'm like 20. And I think that the oldest I am is with some epigenetic law. It puts me at 42, right? So where am I? Like on average, I'm like 32, which is not so bad, but 20 to 42, that's like, if I only did one of those clocks, I didn't have any information. You see? Like that's what's difficult about this thing.
It is very difficult for consumers. And so I think there is some good progress in with this biomarker consortium of biomarkers where they're developing this non-biased cohorts where all of the clocks get tested, and then you select they put out the best performing ones. So in the next couple of years, even if they're starting with epigenetics, then they're going to go into other omics. I think maybe this is within a year's time, you'll be able to go to this and see, okay, what is the model behind this clock? How can I commercially access this clock? And then I would say always go for the clock that's most has the most scientific evidence behind it. And every company.
Does it even make sense from like a longevity perspective to take the average of every clock that's out there? Just like compute all 20 of them and take the average. Does that make any sense, or is it even more useless?
Not at all, because a lot of practices do that, but taking this medium, there is no good evidence to show you that it's actually a good medium, because all of these, again, these clocks are different. I think there's a nice model, like epigenetic model, the denodium pace, which is an amazing longitudinal cohort, which doesn't give you an age, it just gives you a rate of aging. That is both the same clock you have in research now applied to consumers. So I would say if I was choosing an epigenetic clock, that would probably be the one that I would look at and play around with. But apart from that, you can model aging on many different things. And this phenotypic aging, or something that was developed into an epigenetic clock, the pheno-age, is something you can just measure at your routine. Even you might even get these bloods routinely done with your physician, and you can just take the results of these bloods, put them into an algorithm. You have a few of these online, and it gives you an age which is more phenotypic. It's more like if you're ready. These are traditional biomarkers, so they'll tell you when a certain organ doesn't function so well. So they'll like if you have this dysregulated glucose, this will come up quite high, and you will come up quite old unless this is medicated well. And then you have these earlier molecular level clocks. So we would function a much earlier level to that. But there's no reason not to look at both. Both gives you good information.
Okay.
And then there's the lipid clock, which is not yet commercial, that Max is working on. But there is actually a company doing some lipid clocks, but I don't know much about them. Max would know more.
Well, I would say, I mean, if methylation clocks are like early lipid clocks is like where methylation was 15 years ago. So I would say that's like all like scientific experiments right now, and it's fun. What is interesting, what I see on my lipid clocks is that a lot of the important lipids are also linked to the immune system, which maybe goes along with what you said earlier. The immune system is important. That's probably like glycanage is looking at the immune system as well. So yeah, there's like at least the immune system has like signals on different metabolites, and that's obviously, like you said, closer to understanding biology, what's going on, than those methylation clocks. But at the end, I think the grand thing will be like combining every kind of omics, being like super quantified self, measuring everything you can measure, and try to build like a predictor on that that gives you precision but also insights.
About the quantified self, I've been collecting data about myself for as long as I can remember, actually. I think I have like 15 years or more of data. And at some point, I got so much into it. I think I was measuring about 200 different data points per day, not including all the blood tests and everything. I just became anxious. Right? It's just like, it's just too much thinking about every single aspect. How do I feel right now? Oh, my finger is hurting. I'm going to have to write this down. And like when I stopped tracking so much and I started to concentrate on goal-oriented tracking, as in, that's my goal. I'm going to track what's relevant for that goal and then do my checkup once a year. I just felt 100 times better. So like, how can you prevent this sort of like, excessive quantifying that just makes people freak out for no reason?
I honestly think this comes down to mental health, because it's kind of like anxiety that you can self-develop. I mean, we develop anxiety over many things. Social media can be one of these things. And that's one element of longevity that it's very hard to research, and it's also very hard to implement. So that's one thing that we leave out. We accumulate all this data about ourselves. We have the perfect diet. We go to the gym once a day. We're like the most healthy person you can measure. But actually, unless you have this good mental stability, well, it's not just there's so many elements to it. I think that's one thing that I often see missing on these protocols. And it governs a lot. If we look at the studies where there's this famous study that looked at all these Harvard students that were followed for a long, long time. And then you looked at what was the most impactful thing for their longevity. And it was the quality of their relationships. So there's these things that have a huge impact on our health and our longevity that are hard to measure, and we don't have any standards for it. I'm actually excited about clocks coming out in this space and seeing ways how you can change it.
Interesting. So anybody in the audience wants to jump in, by the way, guys?
Yeah, I was going to say, we've only got about 5 minutes left. So if anybody in the audience has a question, they can hop in. I think I see Francois unmuted himself, and this will circle us back to the species question. I think he mentioned in the chat.
No, actually, I just moved on. But
Okay.
Around Kyle and Nicolina and Max, amazing. So many stuff to talk about. Even the CRISPR axe, I know exactly which mutation I would modify in myself. But it's about making sure that we are resilient, I guess, at all possible scales, organism, organs, cells, even DNA repair, and so on. So let's go to the technology, because I really wonder, Nicolina, Max, what do you think about what are the most exciting technologies that you will see in the coming 5, 10, 50 years that would actually push the envelope in terms of longevity? And it can be AI, it can be new stuff in biology, but I'm really curious about what you think will revolutionize longevity in the future.
If you like, I can go first, but mine is very biased in a way, and it's not to do with glycans. So I think that men and women care about different things, and women generally outlive men, but they lose fertility much earlier on. So if we're going to fix this problem of longevity, in parallel, we need to fix the problem of fertility. And you have this very interesting, unusual thing where women outlive men, but their ovaries are the fastest aging organ in the body. So if you look at the oldest man to have a child was 92, the oldest woman to get naturally pregnant was 57. But in average, this would decline from your mid-30s. So I think there's different goals that need to be kind of pursued at the same time. And in this one, there's a very nice animal model example. And this is the first one. Well, of course, you have a biotech company called Fauna Bio that's generally just looking at long-lived animals and figuring out if we can find drug targets for that. And the one that applied most to fertility was this I think it's a naked mole rat, if I'm not wrong, but one of these type of mice that live underground. And it was the only species where they found that they can create more eggs in later age, because all female mammals are born with a set of eggs, and then this is exhausted throughout life. So we're born with about 1 million. By teenage, we have a couple 100,000 left. By mid-30s, we have maybe 25,000, something like that. And then you have an animal model example where they keep recreating these eggs, same as men, throughout their life. So I think taking some lessons there and seeing how we can, and this is a very long translation, it is quite exciting.
Yeah, I think all this reproductive longevity is like super important, for women, but also I think it will in general shed a lot of light on why certain organs age faster. So it will also be beneficial to all men. Otherwise, I think three things I'm currently having most in my mind, what I think more people probably should like develop and look at. One is, I think, like mitochondrial transplantation. Like we know that our mitochondria get worse with age. Can we somehow take them out, grow them, and implant them again, just like to keep the power plants within the cells going? This is still like very early research in a way, but I think it's super cool. And yeah, might just work. There's like some data out there that it seems to work. The second thing is I think like combinational gene therapies. And that is like, okay, like more like mRNA based, not vaccines, but therapies that are then delivered to specific tissues, and the combination of different mRNAs. And then I think one thing is like, which goes along the lines of let's make the things that we learn from parabiosis. And parabiosis is like when you take like a young organism, an old organism, and you connect their blood circulation. The young organism usually gets older and the old organism gets younger. So whatever we find from that, trying to get that implemented into humans, right? So it goes probably like towards like blood replacement, goes towards lymph fluid replacement, or like cerebrospinal fluid replacement. Taking learnings from there and trying to rejuvenate it in a way.
Very cool. Well, I think we're just about at the top of the hour. We can probably take another question if anyone has one at the tip of their tongue. Otherwise, we can close out. I mean, I guess Nicolina and Max, you both got to give your what you're looking forward to in the next 5, 10, 15 years. Rand, I know you've been reading up on a lot of papers lately. Perhaps you have something that's come across your desk that you're eager to share.
I mean, for me, I mean, plan A is, I guess don't die, right? So I'm just continuously looking for like the new things to try. but my plan B is cryopreservation. And I know it's like sounds funny, like kind of like sci-fi kind of thing, but there is this company now in Germany, Tomorrow Biostasis. They took out all of like the creepy stuff out of it, and they just made it life insurance basically. So you just pay like a small amount every month for your life, and then if you die, they just freeze you, and then they'll see in 1,000 years what happens. So like obviously, that's really plan B, not plan A. And it's not something I'm looking forward to. Definitely not. But yeah, I think my personal concern really is about I want to stay where I am. I feel great right now. Like I just want to stay as I am right now. I'm very happy this way. So like most of my effort is on reducing the speed of aging. I'm not looking into rejuvenation at my age right now.
Fantastic. Well, I want to thank all of our guests, Nicolina, Max, and of course, my trusty colleague, Rand. Thanks for joining. And thanks to all of you for joining, contributing questions. And I know for those of you based in Europe, it's a little after hours. So thanks for bearing with us. This has all been recorded. I'm going to chop it up, put it on YouTube. You can look at it later. If ever you want to get in touch with us, feel free. You can respond to the email in the event app, and you'll reach me directly. And yeah, until our next installment of unit testing, we will see you around on Twitter and LinkedIn and all the other interesting longevity spaces we can find. So thanks a lot. Have a great night.
Fun.
Thanks, everybody.
Thanks.
Take care.